So Monica is an associate professor of biomedical informatics at the University of Colorado Anschutz Medical Campus, focusing on developing software tools and standards for biological data analysis and dissemination. Her expertise includes genomics, biocuration, knowledge representation, data harmonization, and artificial intelligence. She is driven by her passion for using comparative genomics to enhance our understanding of human health and disease. And Peter's a professor and Donald A. Rue Chair in Genomics and Computational Biology, heads a research group focused on developing algorithms and computational resources for genomics. He is a principal investigator of the Monarch Initiative, an NIH-supported project for integrating and distributing cross-species genomic and phenotypic data. Significant contributions from his lab include the human phenotype ontology and the Exomizer suite of tools for genomic analysis. Since last year, Peter has been at the Berlin Institute of Health. So with those introductions, I'd like to turn the time over to Monica. Thank you.
Thank you, Grant. Thank you, everybody. It's wonderful to be here. Thank you so much for the invitation, and we're excited to share with you a little bit about what we do with Phenopackets in the context of the Global Alliance for Genomics and Health. I will start sharing my slides, and if you want to follow along, revisit these slides later, the link is on the chat as well. So let me start. By hiding the floating panels. As Grant said, thank you for the introduction. I'm Monica Muñoz Torres, an associate professor at the University of Colorado Anschutz, and I am the co-lead of the clinical and phenotypic data capture workstream for the Global Alliance for Genomics and Health. I'll tell you more about GA4GH as well as Phenopackets. We'll share a few details about implementations, and then Peter will take us on a tour of software and implementations and just sharing with us a little bit about information on tooling for building, validating, and leveraging Phenopackets. At the end, I'll just give you a reminder so that Beatrice, our fantastic project manager, is happy with me that we are resuming our Phenopackets calls, and so you can join this conversation if you'd like. I think this is one of the groups that would understand what has been happening over the past 2 decades regarding the massive technological changes that have, propelled genomics. And but there are issues that still remain on this, right? So we're trying to do what we can to bridge the gap from the research bench to the clinic, in trying to make sense of how do we make use of this incredible deluge of data, and how can we create bridges between research labs and the clinic so that this knowledge that we discover can be put into practice. And so, as you know, because I am certain that you face these challenges yourself, we still have issues trying to develop the infrastructure and the technology that we need to leverage this amount of information, and to also be able to navigate all the ethical and legal landscape so that we can have, ensure privacy and security of the data. So this is where the Global Alliance for Genomics and Health comes in. The GA4GH was launched in 2013. And it has been working to tackle these challenges. This is an international not-for-profit organization that develops standards and policy frameworks that help us to enable the responsible sharing of genomic and health-related data. And so GA4GH works by promoting the sharing of the data, not just across jurisdictional borders, but also across the research healthcare continuum. And all of us at GA4GH, volunteers and in part of the Secretariat, promote the interoperability and harmonized approaches that allow us to work on this and achieve this collaboratively. And so the first question that you might ask is, what can this global approach to genomic data help us with? And why do we care about sharing of genomic data on a global scale? And so imagine, if you would, if we can create a virtual cohort of properly consented genomic data on a global scale, we'll be able to enable a lot of downstream impacts for both research and for precision medicine. And so we would have the ability to demonstrate patterns in health disease, lead stronger variant interpretations, increase accurate diagnosis, just like we're sharing on this screen, and One great example is how genomic data that was shared on a global scale led to groundbreaking discoveries at the beginning, or in the middle of the COVID-19 pandemic. And even though we have these mountains of genomic data, the challenge remains that the data is spread all over the world, and it just resides in different places. And the answer, to answer the most pressing research questions, we need and we must have interoperability between the computer systems and platforms, between data pipelines and databases, between many different jurisdictions. And we firmly believe that standards are the gateway to help us create this interoperability. Oops, that's the wrong direction. So the Global Alliance for Genomics and Health works to develop standards that are organized in basically these 8 focus areas. I'm trying to find my, there it is. These 8 focus areas here on the left. They're called work streams, and they go from cloud genomics to data discovery. I co-lead the clinical and phenotypic data capture workstream with Benjamin Berg, who's here on the call as well. And Peter drives all of the work that our workstream conducts on the Phenopacket standard. And there are 28, I think, as of now, real world program initiatives that provide use cases and provide feedback that allow us to support the development of standards at GA4GH, and also allow us to adopt and test these standards to let us know how they actually work in practice. And so we call these driver projects. Some of them are highlighted on the bottom right of this slide, and they're intended to represent you, the broader genomics community, so we can ensure that our standards are helpful and are actually making an impact. And they cover a vast expanse of the globe and help us ensure that we're genuinely creating internationally driven standards. This image that I have here comes from courtesy of the Secretary of the Global Alliance, and it shows all of the approved GA4GH standards that are mapped to a learning health ecosystem as you see the wheel go around. And so we know basically that knowledge is implemented into practice. That's our expectation, and this generates new and better data, and this feeds back into the system to generate new knowledge. You can see here each of the little icons for GA4GH shows us standards at each part of the steps, from being able to consent the data that is being donated, standards for the distribution and exchange of genomic sequencing, for the review and policy for research ethics committees, all throughout reaching the research clinician. I'm not going to go into detail to each of these because that's a lot, but there's a little link over here to a video that explains this entire wheel. Focus your attention then on what the clinician, so the researcher or the clinician is doing, with the sharing of data sets, analyzing them, retrieving the data sets, and trying to do this entire wheel of discovery. And as you can see over here at the bottom, we have our focus of today, which is our computable representation of phenotypic data, which is intended to improve the synergy between the people and organizations and the systems that comprise this joint effort to address human disease and biological understanding. And we call that the Phenopackets. And so, you are very familiar with this. Phenotypic features or phenotypes like symptoms and laboratory and imaging findings, results from tests are of high clinical importance, but exchanging them with genomic variation information is often overlooked or simply neglected. And we realized that we needed to be able to use computable representations of these phenotypes at the case level, and so that we could have computable means to relate form to function and dysfunction, so that we could interpret the genomes. And so there have been standard exchange formats, as I'm listing here on the left, for a long time, right? Like to represent genome sequences. But we didn't have something similar to this for phenotypes, and at least not in a way that allows us to constrain the data to a model that's research ready. And so we needed this mechanism to share case level phenotypic information that would allow us to improve the use of only, instead of using only free text or downloading the entire EHR into a PDF. And that's where Phenopackets came in, right, to fill this gap. as an exchange format for phenotypes and environmental factors in a little, literally a little packet of phenotypic data that can be used anywhere and written by anyone. These packets of information are encoded using ontologies, and they can be used to relate a patient's phenotypic profile to model organisms, for example, to help improve diagnostics and to help improve mechanism discovery and the integration of environmental health data. And they allow us to exchange information about the patients, including identifiers, age of collection, as well as details about each of the observed phenotypes, including how I'm listing here things like whether or not a phenotype was observed, when they were first observed, how they were observed, and how severe the phenotype is, and how each phenotype is linked to a patient or to their genomic information, to the samples that were collected, and even when available, to the patient's parents and siblings. And in the stable version of Phenopackets, we include also improvements for the representation of cancer and COVID phenotypes. So I'll get a little bit into the wirings before I hand over, for a few more minutes before I hand over to Peter to give us a detailed about tools and implementations. But the schema provides sufficient searchable information of the data outside of the electronic health record. And so this enables us to capture structured data at the point of care by a clinician or a clinical geneticist. And it also allows it to be shared with other labs or computational analysis in clinical research environments. Phenopackets provide a complete computational object model of the data as opposed to, a relational database schema, table schema. So basically think JSON objects instead of TSV files. And this schema supports the exchange of computable and longitudinal case level phenotypic information, as well as diagnosis and research on all types of diseases. This includes Mendelian and complex genetics diseases, cancers, and infectious diseases as well. And this is perhaps an overwhelming figure, and you can explore this one in detail. But I want to tell you that the schema basically consists of several optional elements, each of which can contain information about a certain topic, like a phenotype or variant or a pedigree. And an element can contain other elements. So, for instance, the Phenopacket contains elements of type individual and phenotypic feature, biosample, and individual elements can be regarded as, think about them as building blocks that you can combine to create larger structures. And so a family describes a group that is being investigated for a disease, a trio or a parent and child. And the colors you see here basically represent the major themes of the elements within the schema. And so in a Phenopacket, you can find several different information. As I was sharing, you can see information. These are some of the examples of the most important Phenopacket elements. In the next few minutes, I'm just going to describe a selection of these. And for the complete schema, you can visit this link at the bottom on the slides that I shared. So, for instance, phenotypic features, right? This is what it looks like in a Phenopacket. And these are typically characteristics that are more descriptive than quantifiable. So you have anosmia, which is the loss of the sense of smell, or fever, or dyspnea, which is the difficulty to breathe. And so the Phenopacket can contain information about an arbitrary number of phenotypic features observed in a single individual, and each of them is encoded with a phenotypic feature element. So a subject will generally be a patient or the proband of a study, and the phenotypes will be the abnormalities that are described by an ontology, like the human phenotype ontology. And each phenotypic feature is defined by an HBO term. An example is shown here with the letter A, which is qualified as either present or B on the left, or as absent, right? And or excluded. And you can have the possible severity of the phenotype, as well as modifiers that tell you about the frequency, as it is shown here on D as quotidian. You have information about onset and information about the resolution, and an evidence code, whereas G can be actually also information. And this is information that can be provided to represent the evidence for an assertion, such as the observation of the feature itself. The example in this panel shows a phenotypic feature that can be understood as severe infantile spasms that first occurred in infancy and resolved at the age of 4 years and 2 months in a child that did not have global developmental delay. As well, you can do measurements, and so this is so this is information about the range, the normal range for platelets. And measurements are going to help us do the representation of quantitative and qualitative information, right? And so in here, we're showing a sample that had abnormally low count of cells per microliter. And this in the Phenopacket will be described by establishing a value quantity as well as a reference range where you can have information about the entire clinical observation as well as a timestamp for it, and then all of the information about the assay. Oops, there we go. There's also like capacity to share information about medical actions. And so on the left panel, we see all of the components of a medical action on a Phenopacket over here. And here an example that was done using COVID. And so there was an implantation of a left ventricular assist device in this year, fictitious 2016. And then the medical history here is important as it represents a risk factor. And there was then intravenous administration of dexamethasone in provision of oxygen by a nasal cannula at an initial dose of 2 liters per minute, and that was later increased to 50 liters per minute. And in general, a medical action consists of one of the 4 options, right? Either you have a procedure or a treatment or a therapeutic regimen or radiation therapy. So this covers pharmaceutical treatments and surgical procedures. So basically, Phenopackets, we truly believe that Phenopackets are capable of benefiting researchers and biologists, physicians alike, and they can support diverse users and use cases in the context of this biomedical ecosystem that we're drawing here. And there are multiple providers of phenotypic data that includes you, the researchers, and patients and clinicians, having a variety of mechanisms that include mobile health gadgets at the electronic health record. And the Phenopacket schema basically acts as a common model that can capture data from many sources with a unified software representation, and then can in turn be used by multiple receivers of the phenotypic information, which includes journals and databases and registries. And their implementation can provide the speed and accuracy of diagnosis as well as treatment effectiveness, and can facilitate cohort identification, patient matchmaking during clinical trials, and just in general improve the data landscape, facilitating the creation of tools and algorithms that as they start to find their way into databases and web-based tools. So oh, come on, little thing. This is just an example of some of the production systems that are implementing Phenopackets. GA4GH is an open source organization, and all these standards are freely available. So that means someone can go and adopt our standards, but we wouldn't know about it. So these are the ones that we know are implementing, and they are not a necessarily comprehensive list. If you are meant to be in this list, but you aren't, let us know. And I want to highlight that these are confirmed implementations. There are other organizations that are trying to get into this. If you recognize any of these as an organization that you participate in and you don't know much about Phenopackets, please make sure that you reach out to them or to us. And with this, I'm going to hand it over to Peter so that he can tell us about software and implementations. And so, Peter, I'll stop sharing and you can start.
Thank you so much, Moni. Should I share the slides from my side? That might be easier.
Yeah, cheers.
Just open this. Sorry.
While Peter's doing that, let me say quickly, once again, thank you, Moni, and she was very kind to put the link to her slides there in the chat. Also, you see in the chat that Beatrice has put in some contact information to learn more about GA4GH. And also, if you have any questions about what Moni shared, you can do that in the chat too, and we'll cover those questions after Peter's presentation. Go ahead, Peter.
Are you seeing my slides now?
Yes, we are.
Super. Okay. Well, thanks very much, Moni. I'm gonna talk about some applications for Phenopacket. So assuming you go through the bother of coding your data with Phenopackets, what do you get? And I'd like to say, well, this is kind of a young ecosystem. So Phenopackets were published in 2022, and I think we were happy that a number of groups have started to use this for diagnostic software. I think Moni showed some of them in the previous slide. But I think it's still getting started. And I'd just like to tell a story from 2010 when my department head, Stefan Mundlos, got what I believe was the first genome analyzer in Europe, so the sequencing machine. And we very naively thought that we would sequence a patient with some suspected new genetic disease, find a disease gene, and publish in Nature Genetics, and it would just be easy with this wonderful machine. And so we sequenced and we didn't even think what would happen after we sequenced. But at the time, the Illumina software gave you an Excel file, and there was no software in the world that knew anything about this Excel file. So a gifted postdoc in my group, who's now a professor, Peter Kravitz, kind of went through the file line by line. And after nearly 3 months, he had reached the bottom of chromosome 1 and saw a variant in a gene which, because of some details that I'll skip, was obviously the right disease gene. But that was 3 months. We could do this now in an hour. And I think the current state of using clinical information for bioinformatics is kind of like it was back in 2010. There are ways of organizing the data in FHIR, for instance, but it's not really research ready. It's not AI ready. You always have to massage it. You have to filter it. You have to change ontology terms. And there's a heck of a lot of data out there available in sort of boutique databases. Each of them is different. And so if you're going to analyze that, the first thing you have to do is convert your data. And for each analysis program that you want to use, you have to convert your data into yet another format. And so that's the niche that Phenopackets wants to fill. We want to be research ready, AI ready data about individual patients, often, but not necessarily including genetic data. And so just briefly, where is this data coming from? Well, a lot of the data that's out there is now available in ontology terms. And people have asked me, well, isn't that enough? Why do you need a schema too? Well, a schema is just sort of like, where do I put the ontology terms? Otherwise, if you send me a list of ontology terms describing your patient in a Word file or in some Excel file, I'll have the same problem as I just described. This might be stating the obvious to this cloud, but a schema defines the structure of data and specifies rules and constraints on the elements. Many of these are ontology terms, but as Moni presented, some of them might be numerical, for instance, the platelet count. And so at a very basic level for genetics research, if you use HPO, a phenopacket can be used as a way of arranging HPO terms plus genetic variants plus your disease diagnosis, and optionally even representations of treatment and time course in a registry, although I haven't seen that yet, for analysis. And the main things that you can do now, we think with phenopackets are, number one, you can feed software that does genomic diagnostics. Show you an example. I'm going to spend a little bit more time today to show some software that we've been working on for genotype-phenotype correlation analysis, stratification, a new project in oncology. I'm going to mention the Beacon V2, which was a great GA4GH collaboration with Jordi and others. I don't think I have a slide for the FHIR, but Moni, we should definitely mention the FHIR project before we stop. So just briefly, some of you might know Exomizer. So if you're using this, you collect, you say, what are the phenotypic abnormalities in my patient? You find HPO terms that match them, and you kind of give the program a path to your VCF file, and you give it a list of HPO terms. And when we published this in 2014, we came up with a bespoke YAML format. And so, but this was the only software in the world that used this format. And when we wanted to compare our software to some competitors, we had to reformat the input file and run it again. And so at a simple level, a Phenopacket is just an agreement as to what the input format should be for diagnostic software. And we know of two other pieces of software for diagnosis that use Phenopackets. I think many of the other companies and academics who write this kind of software are starting to support this. Here's a project for genotype-phenotype correlations. I'm going to tell you another story about how hard this used to be. When I was a young lad, and I'm not going to reveal my actual age now, but when I was just getting started in research, I had a wonderful mentor, and she gave me an assignment of analyzing genotype-phenotype correlations in the genetic disease called neurofibromatosis type 1. And that's a very variable disease. I'm sure you recognize some of the manifestations, cafe-au-lait spots, these little benign tumors called neurofibromas. And her hypothesis was that mutations in different parts of the gene would be differentially severe. And, NF1 has about 100 different clinical manifestations. And so my mentor thought that maybe, certain kinds of mutations, like nonsense mutations, are more prone to give you cafe-au-lait spots than other mutations. So that's a genotype-phenotype correlation. And she basically said, here are the charts. So they were paper charts. This was in the late 90s. And there's a computer with Microsoft Excel. And so I started to do this by going through the first chart, and I would put in, a German, or I forget if I was doing it in German or English, the manifestation, and then I would put a yes or a no and so on, or put a number. And then I would get to the second chart and realize, well, this is a synonym. I guess this is the same thing. I'll just put it there. And this kind of kept going, and you realize the data had been sort of recorded in different ways. You didn't always have an asserted exclusion. So if, for instance, if they didn't mention cafe-au-lait spots, was that because the patients actually didn't have them, or did they just not find time to write it down? And anyway, after about 3 months, my Excel file had gotten pretty big, and it got corrupted. And those of you who are of a certain age and were using Microsoft products in the 90s might remember that happened pretty often. And you saw this big red X, and essentially there was nothing you can do. And no, I did not have a backup copy. And my mentor was extremely nice to me, more than I deserved. And she said, well, let's do another project, don't worry. Otherwise, I wouldn't be here. But since then, I've wanted to do a better job at genotype-phenotype correlations. And if you are in human genetics and you look in the literature, what you'll see is that essentially everything that's ever published has some supplemental file, and it's Excel, and each Excel file has a different format. This data cannot be reused. And so you have all sorts of studies that have some small cohort. They're testing different things, and you cannot easily combine those data because they're just not in a shareable form. So Phenopackets can take care of the sharing and can help with recording the data in a standardized way. And this is what we're doing with this. So to do this, we basically curated, it's now 8,000 individual case reports from about 600 diseases, 500 genes. Yes, especially I'm obsessive compulsive, but a bunch of people have been doing this for about 3 years. And now we can take these cohorts of patients and look for genotype-phenotype correlations. And so what the software does, it takes a collection of phenopackets. So this might be a cohort so we did do NF1 actually. And we collected about 400 case reports from the literature. And the software essentially does some quality control, and it shows you a graphic of the location of the the mutations on the protein. It shows you tables with the most common variants and HPOs. It then allows you to divide your cohort up dynamically into 2 groups or even 3 groups according to genotypes. And I'll explain a little bit about that in the next slide. And then it allows you to do 4 main types of statistical tests. So the first one is categorical. So if A here, the dark red, is missense mutation, and B, the pink is nonsense mutation, you're comparing the proportions of patients with missense and nonsense who are noted to have HPO terms. And here we see seizure, freckling, broad philtrum. And so in this case, 30 of 35 patients with genotype A had seizure, and 0 of 10 with genotype B had seizure. So this is essentially, it's like a chi-square test, but for reasons we use a Fisher exact test because that's a bit more robust. If you have numerical values like the platelet example that Moni had, you can do a t-test. A lot of people in genetics make ad hoc phenotype severity scores where you essentially get some numerical score of, let's say, between 1 and 5, depending on how many different manifestations you have of some disease. So that's a U-test. And we can also compare survival. So basically either the age of onset of the disease, the age of onset of a specific manifestation of the disease, like kidney failure, or of mortality. And so this is how how the software helps you to test different genotypes. So the software has predicates. So for a predicate is just a function that returns true or false. And so the predicate takes as input a variant, and then it returns true or false. So you can do this for, let's say, a missense variant. You can do this for a specific variant. You can do this for a variant that's located in a specific protein region. You can look at variant effects, such as missense, nonsense, structural variant, and some other things like that. But you can also combine these in very expressive ways. And so, for instance, if you want to say, compare missense variants in exon 8 against nonsense variants, I just made that up, but there are many examples of things like that do make sense in genetics. You can essentially just combine 2 predicates using an AND function in the software. It's very easy. If you want to compare loss of function variants, well, there's a lot of different types of variants in genetics that can cause a protein to just lose function. Stop gained or nonsense is the most commonly recognized one, but you can also have loss of a start codon, or you can have a frame shift and other things like that. And so you can just OR these predicates together according to whatever your definition is of what functional loss is in your gene. You can also say, for instance, a structural deletion. Well, actually there are different thresholds in use in the field. I think the one that's most commonly accepted now is a change of a length of 50 nucleotides or more, but I can remember not too far ago seeing a definition where this was 2,000 was the threshold, and that might make sense for your research question. And so you can just adjust this simply using this software. Once you have that, those are individual variants, but you then have to create. You then have to classify patients according to genotypes. This is relatively easy in autosomal dominant because everybody just has one variant. But for autosomal recessive, well, you can have, let's say, 2 missense versus missense plus nonsense, or nonsense plus nonsense, and so on. So that it's a little bit more complicated, but the software does all of that for you. And so this just shows some examples. So in panel A, the software kind of creates a cartoon where you see the variants, and this helps you with a couple of things. So it's not good in statistics if you test everything. It's not good science to not have any hypothesis at all. But also you'll get a multiple testing burden. And so we basically try to help the user to pick hypotheses that make sense. And looking at this cartoon for the neurofibromin gene, we see that there are some variants that are very common in our cohort. So the lollipops, these circles and lines, the bigger they are and the higher they are, the more variants of that type that we're seeing in the cohort. So one hypothesis might be that this really common variant might be different from other variants. So you might test that. Another one, you can see the variant effects. So we have missense are the red, but frame shift are the kind of brown. And so missense are more common, but there are enough frame shifts to have statistical power. So it looks like there are about 80. And so another hypothesis might be that that frame shifts are different from missense. Finally, you can look at the protein, and I'm not sure if you can read this, for instance, but you can say all of the variants in a specific protein domain might be associated with something. In this particular case, there's a grouping of missense of variants here that in a region of the protein that doesn't apparently have a recognized domain, but you get the idea. In other proteins, they do. Anyway, and so if you're doing this categorical test where you're basically just testing an HPO term against genotypes, in this case, we're testing the mutation of not just there's actually several mutations that affect the arginine at position 1830. I think that's this biggest lollipop here. And we're comparing it against other mutations. So this little C here means complement. So it's everything else, basically. And you can see that 76 of 133 patients with arginine 1830 mutations, that's 57%, had freckling. So that's a manifestation of neurofibromatosis, like lots of spots in your armpit, for instance. But 90%, 200 of 222 other mutations had freckling. So there's a much higher proportion of freckling if you do not have this mutation. A mutation at this position. And in fact, this is known to be a mild mutation. And we also see that two specific kinds of freckling are reduced, as well as a brain tumor called astrocytoma. This is a little bit less reliable because we don't have a lot of information about astrocytoma in the other mutations. This is because the way that the phenotypes were recorded in the publications was sloppy, and they usually don't write down everything that they excluded. I used to see patients with this disease. You will almost always send these patients to MRI of their brain every couple of years. So they almost surely had this information, but it wasn't recorded. And that's a limitation of this kind of analysis with published data. Here's a phenotype severity score. It goes from 0 to 5. Basically, in this particular disease, that's the number of features you have in 5 different organ systems. I forget what they were, but it might have been brain, heart, liver, kidney, and liver. The de Vries score is named after the Dutch geneticist Bert de Vries. I know him, he's a nice guy. And he came up with a slightly more complicated score that just counts manifestations in a more detailed way. And this is another disease. And in this case, missense mutations are milder than non-missense mutations. This is a t-test for 17-hydroxyprogesterone. That's a numerical level. And this is another disease where a mutation affecting a cysteine residue at position 248 and turning it into a tryptophan is very severe. So the onset of stage 5 chronic kidney disease, basically meaning you need dialysis, is at an earlier age than with a mutation glutamine at position 316 to proline, which is the pink line here. And so this, basically it took me 3 months to fail at doing this for one gene. Now with this software, I can do this kind of analysis for one gene in an hour or two if I know the gene. Obviously, if I have to read papers about it, it'll take longer, but it turns what used to be an incredibly tedious and horrible task into something that is feasible if you have your data as phenopackets. And it also means that if you have something in your center, I have something in my center, as long as we agree to collect the same data, it's child's play to combine our data to get greater statistical power. So there's a lot of advantages to doing this with Phenopackets. Moni, you'll tell me if I'm running out of time, please. But
Cheers. We have 15 minutes left on the hour, and we want to make sure that we get some questions. So whenever, yeah, you take the turn.
okay, so maybe I'm going to go quickly. So I'm really excited about the genotype, about this correlation project. We have a similar project that allows you to cluster patients with phenopackets using a new algorithm. Can present that another time. This gives you an idea of what it looks like, and in this particular case, we could stratify a cohort into 3 groups. We were using basically the same phenopackets, and our gold standard is just the diagnosis of a certain gene. Not because that's scientifically interesting, it's just a good gold standard for an algorithm. And we are actually able to recapitulate the stratifications that we see in OMIM. And we have used this, a very similar algorithm once to look at long COVID and found what we think are generalizable subtypes. So we're happy to share these slides so you can look that up. Another new thing, and this is unpublished, and this was led by my great colleague Justin Rees at Berkeley. And we basically were working with people at the NCI to create phenopackets for cancer, and so phenopackets also have a lot of slots essentially to represent a biopsy that can be from a tumor or an extirpated tumor, and in this case, we were representing somatic, and this is just one of many examples that are now available via this resource, but we were looking at somatic variants in a gene called IDH1 in glioblastoma. This is very well known, and we just used this as a gold standard, but we could start from the phenopackets and again do a survival test. It's essentially the same computer code as we had in the other program. And again, this will allow data to be exchanged. Previously, everybody who had cancer data had their own format, and the same problems pertain. Just like to mention that Phenopackets are compatible with the beacon interface of GA4GH. Jordi helped us an enormous amount to get this to run, and we had, as a pilot project, we basically put a beacon API on top of the Broad Institute's Terra system, and allowing you to query a very small set of prenatal genomes. That was just a pilot project, and we are now looking for funding to expand this a little bit. Last slide. If you liked this talk and you know somebody who's looking for a job in bioinformatics, we are hiring. I've got 4 open positions and the deadline is February 14th. We will probably have more positions later in the year as well. So we're looking for good people who are interested in topics like this. Thank you for listening.
Thank you, Peter.
Yes, thank you very much. Really interesting. And even though I've been a participant in this project on the G4GH side, I learned quite a lot of new things from these presentations. Once again, if you have questions about what you saw, please put them in the chat. But you had mentioned quickly, Peter, that you wanted you and Moni to talk about the FHIR versions of this project. Do you want to share that quickly?
So, Moni, should I, or do you want to?
Go ahead. I'll try to find the link, but yes,
Yeah, I
this
mean,
is.
so does this audience know what FHIR is?
I don't know for sure. So why don't you do a quick summary?
So FHIR is essentially a computer interface that lives between your code and an electronic healthcare record system such as Epic, Cerner, or many others. And the reason that this is important is that essentially every hospital has a different EHR. It's set up in a different way. And so if you want to run analysis software on it, again, you would have to adapt your software for each hospital. FHIR essentially is an adapter that provides a standard interface to that. FHIR is a standard of the HL7 community, which is responsible essentially worldwide for all EHR standards. and it's a very flexible standard. It's a schema. It's roughly speaking, it's like XML. So you can do anything, almost anything in Fire, and this means that if you're getting data that's in Fire, it might not have exactly the same structure if you're getting it from two hospitals, despite all of the standardization. For that reason, we want to apply more constraints to Fire data. That is the purpose of something that is called an implementation guide. That's an additional set of rules, and that basically tell you how to implement a Phenopacket in Fire. And this is now going up for a vote for balloting by HL7. I should say this work was led by my colleague Melissa Hendel and Shahim Said in her group. But many other people have contributed to that. And we think that it might help to sort of anchor genomics into the EHR and also to enable more streamlined output of Phenopackets from EHR to sort of go via this this implementation guide.
Moni, do you have anything to add to that?
No, thank you, Peter, for sharing that. And I pasted the information about this is called the Phenomics Exchange for Research and Diagnostics is the name that we have for the implementation guide on the HL7 website. So I pasted that on the link. Thank you, Peter. There's a question from Ben, Peter,
Yeah.
that I think would be brilliant to go over.
Go ahead. Yeah. Moni, why don't you go ahead and read the question?
Benjamin, if you want to share it yourself, that's possible too, since you're here.
Sure. So curious about the differences between Phenopackets and OMOP, particularly in terms of computability. And also if you could talk a bit about what it takes to convert from OMOP to Phenopackets, whether it's kind of straightforward or whether there are a lot of nuance.
So OMOP is similar in some ways to Fire, but it's intended to be sort of a research only system. So it's not part of the live EHR system. It's kind of like an image of the whole system that's been converted into a set of tables. It is possible to export from OMOP to Phenopackets. A group in the US published software to do that, a set of scripts. And so this might actually get you far enough to do Phenopacket-driven research with this. However, at least the use cases I presented so far in rare disease require that you have sort of rare disease information in the OMOP system, and currently there is not only a relatively small portion of the HPO is available for mapping via OMOP. The OMOP team has, and this is Andrew Williams at Tufts, has told me that they would like to essentially ingest HPO into the OMOP universe, and this will be based on work by Tiffany Callahan, who was at Larry Hunter's lab. I'm trying to give everybody credit where it's due, but just to emphasize, it's not just me, certainly it's not even just my group. This is like a ton of people working on this stuff. But so there are reasonable ways of doing this right now, but the community is now trying to more deeply integrate HPO into OMOP, and then using the software that was published by the other group, where it's not, you know, there are various ways you can do this. You can create Phenopackets. Well, I mean, one of the big challenges is just the kind of the philosophy, because what we consider to be a phenotypic feature is not well represented either in Fire or in OMOP, that essentially have either things that are regarded as problems that are called observations, for instance, and things that are regarded as measurements that may or may not be important. And a phenotypic feature in our universe is kind of it's something that in some ways could be relevant, but so it's kind of sits between a measurement and an observation in Fire speak. I'm sorry if that got to be too tedious and dry. It's not something that, yeah, just do it and you'll get everything you want. It requires thought and work to get.
Yeah. And OMIP is, I think I've heard it referred to as this before, as a nice jackknife, because it actually mixes structure and semantics. But the Phenopacket gives you a little bit more of the expressivity of the information about the phenotypic features and a few ancillary, more information. And you were asking, Ben, is it a straightforward, and as Peter has defined, not yet a straightforward mapping, but Peter, maybe something we can consider with some type of mappings to assist for the conversion.
We should invite that other group to our meeting. I'll send you a link.
Okay.
Just came out like 2 months ago. Who made this OMAP to Phenopacket converter?
Yeah, with Nuria. Yes, I remember. Eh, I see some thank yous on the notes, and I didn't see other questions, but I just wanted to make sure, as Peter was saying, giving credit where credit is due, and I just wanted to kindly thank and acknowledge the work of many people who are behind everything that we have been talking about today. On the Global Alliance for Genomics and Health side, we had from the Secretariat, Beatrice Amos, who's been supporting our work, and Lindsay Smith, who is now with Sick Kids Canada, who used to be one of the engines behind our coming together to work on this. And there are many GA4GH contributors, as well as as implementers, and all of the people mentioned here for their contributions. So I just wanted to make sure that we shared our little thank you note before we parted.
Could you indulge me just in the last minute or two, both Moni and Peter, if I were to ask a very practical question there might be people on this call that are hearing about Phenopackets for the first time and going, wow, this is very powerful, but they would have no idea where to start. And I know that's a huge question, but in a minute or two, could you quickly describe what type of a team that a implementer would need to have, and just quickly describe what skill sets that team would need?
So I could talk about our experiences. So I mean, it kind of depends on what exactly you want to do, but there are resources in both Java, Python, and Rust for working with Phenopackets that our group has published open source. An important thing is quality control. It's in the early phases of this project, I determined how easy it was to make lots of mistakes, and so you need some software to kind of check your work. It turns out on a technical level that code to create Phenopackets can look verbose and scary, and so it's often nice to have essentially wrapper libraries that hide some of the complexity and sort of are guaranteed to put the stuff in the right place. We have code like that for both Python and Java, and there is code that essentially starts from text mining and goes to a Phenopacket. One of them is called Phenopacket Lab, that's available as a demo in the cloud, but you can also set up an instance in your hospital. If you are interested, we'd be happy to kind of advise you on the software that is available. Moni showed a slide, so a lot of people have kind of set this up in their own way, and so as always in software, there's more than one way of doing it, but we'd certainly be happy to share how we've done it.
Any quick thought, Moni, from your perspective?
Yeah, no, I think that Peter said it all. It's important to figure out first and foremost what is your need, right? And once you have assessed that landscape, then you'll be able to figure out what are the best ways to address it with the personnel. Obviously, having someone who has some well-versed experience on software engineering will be very very useful in order to be able to support your implementation. I always think that it's very, very critical to connect with someone who is your end stakeholder so that you can actually figure out what's the best way to represent the data that you need. If you're in a clinical genetics laboratory trying to diagnose, there will be certain needs that you will need to address, and Phenopackets can actually help you with the representation of information that you are accustomed to entering by hand. And you should be able to ask the person on the receiving end of this information, what is the goal, what's the best representation of the data that they want to receive, and that way you can have a better estimate of, how big is this task going to be, and what are the personnel that I'm going to need to fulfill it.
Fantastic. Thank you both, Moni and Peter. This was an excellent presentation. I think that our people who joined today got a lot from it. And once again, if you want more information, both Moni and Beatrice shared links in the chat about how to contact a GA4GH and to learn more. So with that, I will thank everybody. We'll just quickly turn the last 30 seconds over to Latifa, who will tell us about the next webinar.
Okay, thank you, Grant. Thank you, Moni and Peter, for this insightful presentation, and you all for your participation. Yeah, so we are excited to feature Dr. Ben Basby from DNA Nexus as our guest speaker for our next webinar that is scheduled for February 27th at 9 a.m. EST. The title of his talk, I've shared it with you on the chat box. It's about how biobank data is changing the landscape of statistical analysis and so forth. And please stay tuned for the webinar invitation so you can register and attend this exciting talk. Thank you again, and looking forward to your participation, and have a nice day.
Okay, bye.
Thank you. Thanks for the invite.
Bye.
Bye.
You're welcome. Thank you, everyone. Bye-bye.